Today's video is our introduction to topic 4.1, Intro to Ecology. This goes along with the IB SL reading guide for 4.1's part 1 and part 2. And when we open up for ecology, the first couple slides here are all about the understandings, which are that's hopefully self-explanatory, but these are the concepts that come out of topic 4.1. Some of these go along with the self-check questions, some of them are definitions, but it's really just the basics of an introductory chapter on ecology. And then the third slide in today's presentation here are the skills. And we can see in the skills we've got putting organisms into specific categories based off of their mode of nutrition, setting up a sealed mesocosm. We will set up mesocosms in class. We'll talk about what those are. And then we've got some math skills here. We've got a test for the association between species using a statistical test known as the chi-square. We're also going to recognize and interpret the statistical significance of our results. And then another skill we'll have is doing some quadrat sampling, a classic field study method to help determine the quantities or densities of species in a given area. And that's really a great tool for ecologists to use, a very common tool for ecologists to use. When we talk about ecology, the first thing we have here is a definition of ecology itself. And ecology is all about the relationships between organisms and their environment. Okay? And the environment can be made up of both biotic and abiotic factors. A couple of new vocab terms here as well. Bio means life, so biotic are living things, and abiotic are non-living things. And our flowchart here takes us from the population level, which is a group of organisms of the same species. We'll do a little bit more with this, right? Same organisms in the same species can interbreed and produce fertile offspring. Populations, and then we have the level of community. And then when we have the communities, which are populations, again, living things and their habitat, again, that could be living and non-living, that's what makes up this ecosystem here, okay? For some definitions we've got to have, the first is a species. So the biological definition of species is organisms that can interbreed and produce fertile offspring. And the idea here is, in this case, we've got an image here of a mule. That's the cross between a horse and a donkey, two closely related species. But when they interbreed, they produce a hybrid, and that hybrid is sterile. Okay, can't produce its own offspring. And the reasons for that can be genetic, for sure. There's genetic speciation. There's also temporal, right, time speciation, right? Two organisms or groups would have to be in the same place at the same time. There are anatomical barriers that separate species, behavioral, etc. So all of these things can lead to individual species. But the key here for species, organisms, they tend to look the same or similar, and they can interbreed and produce fertile offspring. The next level up is populations, and the key for populations is it's the same species of organism. It's a group of those organisms that are now in a specific place at a specific time. And again, in the image in the back, there's a population here of zebra, right? They live in the same place, they are here at the same time, and that's what makes them a population, okay? Again, we've got species that are, again, we see geographic and temporal, right? So space and time that are unlikely to breed. They would then be considered separate populations. You could have a population of fish in Lake Audubon. You could then have a same species of fish, a separate population in Lake Fairfax, right? Something like that. The image in the back is not just zebras, right? There are multiple populations here, and that would be the level of community. So at a community level, now we have populations, and again, those populations interact and are in the same area at the same time. Not just animals, but also plants, microbes, right? So bacteria, fungi, all of that is part of the community that we see in this image. When we talk about the interactions of these organisms, oftentimes we're going to talk about feeding, right? And we really have two big categories of groups when it comes to feeding. We have autotrophs and we have heterotrophs. both autotrophs and heterotrophs need organic molecules. Okay, so all of our biochemistry is based off of organic molecules, and the energy that we need to do work, ATP, another organic molecule, is generated from organic molecules that we burn. So we need that for metabolism, growth, reproduction, all of the life functions here. Okay, for the definition of autotroph, which we often think of as green plants, and we say that all plants are autotrophs, that's not entirely true. Okay, so just be careful there. Autotrophs, by definition, are able to synthesize their own organic molecules. Okay, and that's what makes them producers. But not all plants can do that. Okay, but photosynthetic autotrophs take in the sun's energy and produce organic molecules, and are therefore what most plants fall under. Okay, heterotrophs, on the other hand, obtain their organic molecules from other organisms. So whether it's a plant eater, a meat eater, some combination of both, a decomposer, they are considered heterotrophs because they're getting their organic molecules from other organisms. When we talk about those autotrophs, the classic, right, plants, photoautotrophs, they're able to convert carbon dioxide and water to inorganic molecules into organic compounds. Okay, and then once plants make those sugars, they are then able to convert those sugars into all the other organic compounds that they need: starches, other large saccharides, cellulose, fats, lipids, amino acids, etc. Okay, so those are photoautotrophs, and for photoautotrophs, their light energy is their source of energy to power up those reactions. There are chemoautotrophs, right? Another autotroph, so again, able to convert inorganic material into organic, but they're going to use the energy found in the bonds of the inorganic molecules, like hydrogen sulfide. We've got some tube worms at the depths of the ocean at these sulfur vents that are able to do autotrophic nutrition without exposure to sunlight. We also have, so we have the exceptions to the autotrophs, right? And that's what we see here. Sometimes we get a mix. This euglena here is both photosynthetic and heterotrophic. It is able to do photosynthesis, right? So it's got the ability to convert light energy, inorganic molecules into sugars, but also it takes in food particles by phagocytosis. So it does, it's heterotrophic as well. Okay, that's an example of what's referred to as a mixotroph. A Venus flytrap is another classic example there. So Venus flytraps live in an environment that is poor in nitrogen and some other molecules that are needed. And basically they are photosynthetic, right? They are green plants, but they capture insects, spiders, other small organisms, and decompose them to get the nutrients that aren't available in the soil. Cool adaptation for both of these, right? Okay, so those are autotrophs, and then we have some chemoautotrophs. Under the category of heterotroph, we also have the decomposers, right? And for decomposers, we have detritivores that we see here. And detrivores are internal digesting decomposers. So detritivores take in non-living organic material, right? So material that was perhaps once living but is no longer. They digest it internally to get their energy. Earthworms are classic there. Woodlouse, roly-polies, some other organisms that do this, but these are internal digesters. In contrast to that, we have saprotrophs. So saprotrophs are external digesters, and again, decomposers break down other organic organisms for their nutrients, but they do external, so they secrete enzymes into the environment, and then they absorb those back up. And we see some mushrooms here, some classic examples of saprotrophs. So saprotrophs, detritivores are both heterotrophs, special classes under that category. Okay, and we see a flow chart here. Here's our heterotrophs on the right, which again includes our decomposers, both saprotrophs and detritivores, and then the classic consumers, right? Things that eat other organisms that are either living or recently killed. Humans fit in the consumer category here as either primary or secondary consumers, right? So we have the teeth to be omnivores. Some organisms are carnivores, some are strict carnivores, some not so strict. And then we have the herbivores that eat plants. So the consumers and the decomposers make up heterotrophs. And then the autotroph side, again, we've got our photoautotrophs, green plants, phytoplankton, algae, like the classics. And then we've got these special class here, these chemoautotrophs, like these chemosynthetic bacteria at these deep sea vents. Okay. When we talk about all of these classes, whether they're autotrophs or heterotrophs, any of the classics underneath, they're all going to interact with each other, and they're going to interact in this level of ecosystem. And this ecosystem is both those living things and they interact and how they interact in the abiotic environment. So in our image here, we've got this coral reef ecosystem, we've got water, which is abiotic, we've got sunlight, which is abiotic, and those impact the living things, the corals, the fishes, the jellies, etc., that are here in this image. We're going to talk about cycling of nutrients, right? When we talk about these feeding methods, we're talking about the intake of food. That's all about the transfer of nutrients from one organism to the next. We'll spend some time here. And then we'll talk about the flow of energy, and we'll compare nutrient cycling to energy flow when we get a little bit further into ecology here. To help us study these things, we're going to set up these mesocosms, right? So these mini ecosystems, we're able to test the factors that impact the stability of these ecosystems. Ecosystem stability is an area of great study. So certainly if you have a sustainable ecosystem, it's able to sustain itself, right, indefinitely. But that means we need to have nutrients that are moving from one organism to the next, cycling through. Organisms in that ecosystem need to detoxify waste, and then there needs to be energy available, whether that's for the plant's incoming solar energy, the conversion of that into plant energy that we other organisms then consume to let that energy flow through those ecosystems. Okay. All right. All of these, the first few slides here have all been sort of some vocab, right? Some sort of understandings here. This list of skills is a little bit more detailed than the first slide that we had here. And the skills are where we're going to spend some time in class, like setting up the mesocosms. We're going to work on this chi-square test after we do some quadrat sampling. And you'll develop these skills as we go along. But the slides before this are really just about the understanding, certainly the vocab, and getting through some of those self-check questions. So hopefully this helps you out, and we'll have another one coming up for the reading guide for questions part 2, right, 6 through 10, excuse me. And then there'll be some specific ones on the chi-square testing, quadrat sampling, and then some of the statistical analysis stuff as well.